Hello everyone. So today the topic that we are going to learn is cell cycle. So what is cell cycle? As we all know that for every cell, from every parent cell, two daughter cells or new cells are produced, right? So how are these cells produced? Uh, to form any new cells, we need the DNA or the genetic material, right? So this genetic material that a cell gets in a copy is uh, is through various cycles or various steps, and this cycle is known as cell cycle. So cell cycle is an ordered series of events that occurs. Earlier standards we have studied that the cell cycle mainly consists of two phases, that is interphase and mitosis phase. Now this interface here occurs. In this interface there are mainly three phases: G1 phase, S phase and G2 phase. This G1 phase is mainly for cell growth, S phase is for DNA replication and G2 phase is for mitosis. And after preparing for cell division, in interphase the cell moves towards the actual phase of cell division, that is mitosis, that I would be coming up with a new video. So in this video we would be mainly concentrating on interphase. So if a cell is ready to divide, then only it would enter the mitosis phase. But if the cell is not ready to divide, it will enter the phase called G0 stage. What is this G0 stage? So in this G0 stage, mainly there are three types of stages. First is quiescent stage, second is senescent phase, and third is terminal differentiated cells. Mainly there are two stages only, quiescent and senescent, right? So quiescent state, as I have mentioned, it is reversible phase. What does it mean? That if the cell is not ready to divide and it enters the G0 phase, there are some cells that for some particular point of time it would be in a resting phase, and when the conditions are favorable, then only it would be proliferating or dividing. So this is known as reversible or quiescent stage. But the next phase is senescent phase, where this is an irreversible stage, means that the cells which once have attained the senescent would no longer divide in any condition. Even if it gets a favorable condition, then also it won't divide. So this is a senescent stage. Now this terminal differentiated cell means that as we all know that cell needs to divide and divide to attain a specificity. Suppose from a stem cell or from the cells, if a cell is has destined to be a neuron, then it has to undergo various series of division before attaining its neuron stage. So there are some cells that after forming a particular cells or after attaining the specificity, they stop dividing, means they have lost their ability to proliferate after they have attained specificity, and this is known as terminally differentiated cells. But majorly the G0 stage only has two phases or two stages, that is quiescent stage and senescent stage. Now as we all know that a human cell cycle mainly have the human cell cycle is of 24 hours. In this 24 hours, 95% of the time is given to interphase, means the cell is preparing for the division. As I mentioned earlier that the cell is preparing for division means cell growth occurs, the DNA multiplies and all that thing. So in that, G1 phase has 11 hours, S phase lasts for 8 hours, G2 phase lasts for 4 hours and M phase lasts for 1 hour. Now, what are the changes in the number of chromosome and amount of DNA during interphase? So, as I have mentioned earlier, that in G1 phase or for any cells, when the daughter cells are formed, they need new chromosome and new uh, DNA or genetic material. Of course, then only the cell will survive, right? So for that, in G1 phase, the number of chromosome and that the number of DNA means this 2n indicates number of chromosome and 2c indicates the amount of DNA. So in G1 phase, the amount of chromosome and amount of DNA are both 2n and 2c. But in S phase, as I mentioned earlier, that DNA replication occurs. So in this phase, the amount of chromosome or the number of chromosome remains same, but the DNA replicates, and from 2c its number increases to 4c. After that, in G2 phase also, the chromosome number remains 2n and the amount of DNA remains 4c, and then the cell would move towards the M phase or mitotic phase. Now this M phase would start with nuclear division, means first of all the nucleus will divide, and after that it would end with the cytokinesis, means when from a parent cell the daughter cells are formed, two daughter cells are formed firstly, right? Two daughter cells are formed. They both will receive equal number of or equal amount of genetic material, right? So, and this occurs only in mitosis. In meiosis, equal amount of genetic material is not received. They would receive half the amount of genetic material so that during fertilization the diploid condition is sustained. But in case of mitosis, both the daughter cells would receive equal amount of genetic material, and after that this genetic material has been divided, then the process of formation of cytoplasm occurs, and that is at the end of the M phase or mitotic phase. Now, as we all know that our body is made up of millions and trillions of cells, right? So if these cells would have lost the control, or if this cell loses the control, then what happens? The the cancer occurs, right? The definition itself, what it suggests, cancer definition suggests that uncontrollable division of cell. So in order to control the division of cell, the cell cycle is controlled, so that even if once the cell has attained full ability to divide, then only the cell can divide. If the cell has not attained its ability to divide, till then the cell cycle would be in control. And two major subunits that control the cell cycle are cyclin and CDK, that is cyclin dependent kinase. Now, for every phase of cell cycle there are different cyclins, right? For G1 phase there is a cyclin, for G1 S phase there is a cyclin, for G2 phase there is a cyclin, for S phase there is a cyclin, and for the M phase also there is a cyclin. In this figure we can see that for G1 phase the cyclin is cyclin D, and CDK, that is cyclin dependent kinase, is CDK 4 and 6. Now this cyclin is basically a regulator molecule, and this cyclin is synthesized and divided, means destroyed, after every phase. Means in every phase a new cyclin comes and attaches to cyclin dependent kinase. This CDK is constant in every phase of the cell cycle, but the cyclin keeps on changing. Now for G1 phase cyclin is cyclin D and CDK are CDK 4 and 6. For G1 and S phase the cyclin E is present and CDK2, it attaches to CDK2. For S phase majorly cyclin A comes in picture and it attaches to CDK2. For G2 phase cyclin A is there and it attaches to CDK1. And for M phase cyclin B is present and it attaches to cyclin CDK1. Now, how is this cyclin regulated? As I have mentioned that after every phase this cyclin is destroyed, right? So how, and also the destruction of cyclin for every phase is different, means for G1 phase there are different enzymes that destroy this cyclin. For S and M phase also there are different enzymes that destroy this cyclin, and this majorly occurs through the process called ubiquitinylation, right? So uh, so Regulation of activity of cyclin CDK complexes, right? So cyclin, as I've mentioned earlier, that it synthesizes and destroys. Now in G1 phase, the enzyme that helps in destruction of cyclin is SCF. What is this SCF? SCF means S stands for uh, S, S, S stands for SKP1, C stands for Culin, and F stands for F box. These three units, along with the protein known as a ring domain protein known as RBX1, it helps in ubiquitination and ultimately destruction of cyclin. Next, in M phase, the enzyme that is present is known as APCC, and this also ubiquitinates and destroys the cyclin. Now, this APCC do not contain the F box. Apart from that, the mechanism for ubiquitination is same as that of SCF. Now there are other mechanism of regulation also present, and first among them is phosphorylation and dephosphorylation. What is this phosphorylation and dephosphorylation? So this phosphorylation and dephosphorylation is mainly through a protein called V1 protein. Now what is this? So in order to control the cell cycle, or in order to uh, in order to control the cell cycle, means that cells, if they are not ready for division, or if they are not properly have grown for division, won't go for division. So to control this process, a protein known as V1 is present. Now what happens is that this V1 phosphorylates it is mainly an inhibitor protein, inhibitor residue, right? So it phosphorylates position 14, position 15, that is a tyrosine position of CDK, that is cell dependent kinase, and this stops premature division of cell cycle of cells, right? So in this is majorly in case of yeast. Now in human there is another position, that is position 14, where it phosphorylates. So in human there are two 14 and 15 position where this phosphorylates and inactivates this CDK, so that cell do not divide. Now an antagonist to this V1 is CDC25, means if V1 amount of V1 increases, this CDC25 would decrease. Now when this antagonist work, when a cell is complete and it's mature for division, then only this CDC25 would come and uh, remove the phosphate, right? It would remove the phosphate group from both the position 14 and 15, and that would ultimately lead to cell division. So this V1 is antagonist to CDC25, and when the cell mature, this CDC25 comes in picture and would ultimately uh, help in dephosphorylating or remove the phosphate of position 14 and 15 of CDK, and then the cell can further uh, go for cell division, right? So in this video it's all. Now for the next video I will be bringing is for mitosis. Thank you.